Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Razan, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Charles Meadows from Sartorius-Stedim Biotech.
Thanks, Leah. I'm the product manager for Sartorius's MyCAP CCX cell culture expansion system. And this exciting new product modernizes cell culture operations in clinical, pilot, and GMP production. I'd like to share with you the story of the product's invention and how it can streamline operations, reduce costs, and fight batch failure from contamination. Now, almost every cell cultivation begins with a vial of cells from a working cell bank. The goal is for lots of these cells to grow happily in a bioreactor. But of course, the cells from a vial cannot simply be added directly to a seed bioreactor. In the case of suspension cultures, the cells are transferred from successively larger Erlenmeyer shake flasks until the population is large enough to be added to the bioreactor. At each transfer or passage to a new flask, cells are sampled for total count and viability, fresh media is added to the next flask, and the culture is transferred to the receiving flask, usually by hand pipetting. The flasks have to be opened for each of these operations. Because the flask is opened, there is a real danger of contamination. A Bioplan Associate survey inquired about the frequency, cost, and leading causes of batch failure. On average, a failure occurs every 9.4 months and costs between $1 to $2 million. The leading causes: contamination and operator error. So it was apparent to us that opening flasks during cell expansion is a contributing risk to contamination. And if we could fix this problem by closing the system, we would enable a production process that's less likely to become contaminated. But it turns out there are more benefits to having a closed system. Today, cell passaging is done in a biosafety cabinet. These are expensive to operate and expensive to maintain. The hood and every piece of equipment that's used in it needs to be decontaminated first. GMPs require an environmental monitoring program for the BSC, and most agree that operating outside of a BSC would be simpler and less expensive. And still, despite working in the BSC, many expansion processes continue to have some rate of contaminations. So there is a further strategy to prevent them. Backup flasks are often included at many passages. The purpose is if a flask in a later passage should become contaminated, the operators can use a backup rather than abandon the expansion, which could be days or weeks old. Eliminating backups in an expansion process would save time and reduce costs. These issues of contamination and the labor and cost-intensive process painted a very clear picture for how we can make an impactful change on cell cultivation. Could the expansion process be done outside of the BSC without the risk of contamination? Meeting this challenge would mean a reduction in waste, a lean expansion process, and reduced reliance on biosafety cabinets and the expenses related to them. This is the cap of a typical Erlenmeyer shake flask used today. Notice the large embedded filter membrane in the cap. Cells in a culture consume oxygen and produce carbon dioxide. An imbalance of either will cause growth problems. Adequate gas exchange is critical to successful cell growth. This membrane in the cap is large because gas exchange in the incubator is passive. There must be unrestricted airflow across the membrane to support cell growth. The issue with this cap design is there's no way to get fluid into or out of the flask without unscrewing the lid. Technology of caps with integral tubing is available today. A solution like this allows for aseptic fluid transfer outside of the biosafety cabinet. The filter membrane in this disc filter, it's huge, but the airflow to it is restricted at the hose barb. There is not a large enough volume of air moving across this filter membrane in the passive environment in the incubator to support the adequate amount of gas exchange during cell growth. The novel manufacturing process of MyCap permits us some unique capabilities. Aside from putting tubing in the cap, we can install other components. So we created a gas exchange cartridge. The cartridge maximizes the opening to the filter's membrane and optimizes the footprint, allowing us to include integral tubing alongside it. Our gas exchange cartridge has a significantly larger surface area than disc filters and is even larger than the opening on a traditional vented cap. What we've done with MyCAP CCX is simple. We've combined two technologies into one. We've taken the large open surface area of vented caps and integral tubing of transfer caps to create MyCAP CCX. Once developed, we needed to evaluate performance. So we performed parallel expansions with CHO cell lines on 500 ml to 3 liter flasks. This table shows the culture doubling times for MyCAP CCX in orange and the traditional flask in black. After each passage, the doubling times were nearly identical, indicating there's no discernible difference of growth between MyCAP CCX and the incumbent system. A t-test confirmed there was no statistically significant difference between the growth rates of the two systems. Now, this should be considered as evidence that MyCAP CCX supports cell growth, but we expect our customers will require a comparative study on their own cell lines. To help, we built a tool to assist with the design, execution, and interpretation of results for a validation study on your own cell lines. But even before validation begins, a cost analysis may be necessary. We created a tool which looked at 4 categories of operating expenses in a typical cell expansion process. We looked at cost of consumables, cost of media, cost of environmental monitoring, and cost of labor. Now, as expected, the cost of consumables is higher with MyCAP CCX. After all, the product is introducing tubing, fittings, and connectors. However, all other costs are reduced significantly. Overall, we do expect MyCAP CCX to reduce your operating costs. Our models demonstrate a 9% reduction in costs, but results may vary. We will work with our customers using our proprietary tools to calculate a cost comparison with their specific operations and costs taken into consideration. This diagram illustrates the step for passaging from flask to flask and flask to reactor with MyCAP CCX. Note first that these steps are done outside of the biosafety cabinet. Next, notice that MyCAP CCX uses commonplace parts and routine operations. Peristaltic pumps transfer fluids, balances are used to control the transfer volumes, and connections are made by tube welding or aseptic connectors. In fact, we are not introducing a new flask. MyCAP CCX is currently available on Corning's plastic Erlenmeyer flasks. We are simply introducing an improved closure. A change to MyCAP CCX is a modest and very manageable change with significant upside. MyCAP CCX advances cell expansion operations by eliminating the need to perform operations in a BSC and at the same time reduces the risk of contamination from non-aseptic operations. It was an enabling manufacturing technology, technical know-how, and close collaboration between supplier and users to develop a technology which saves time and reduces labor and waste. This concludes the portion of this webinar. Again, I'm Charles Meadows. I'm the product manager for MyCAP CCX.
Great. Thanks, Charles. So the first question is, what is the void volume in the sampling line?
Yeah, that's a real good question because sampling is a requirement throughout this process. And on these particular designs with MyCAP CCX, we specified very small diameter tubing for the sampling line, so the holdup volume is quite low. On our 250 and 1 liter flasks, that void volume is in the range of 0.5 to 0.6 milliliters, and on the larger flasks, the 2 and the 3 liter flasks, it gets to about 0.9 milliliters. And so it's important that this volume is low, especially on these smaller flasks, because that culture is left in the line after the first day's sample. And that volume needs to be purged before the final day sample is collected. Because there's small volume in that tubing, you're going to limit the total loss, and the purge volume that needs to be done before evacuating that line or to evacuate that line can be as low as about 3 milliliters.
Okay. And do the flasks need to be stabilized in the incubator to prevent tipping?
Yeah, so if you remember, as I noted, the flasks that we use are Corning's Erlenmeyer flask. So it's a standard flask. So the CCX solution will fit into whatever stabilization method that may be being used in incubators today. And there's a number of different techniques. The MyCAP CCX, it is more top-heavy than the traditional flask because of the tubing and the fittings. But the experiments that we've done and also feedback from our customers who are using the product have proven that something as simple as double-sided sticky tape is perfectly fine to stabilize the flask while it's being shaken in the incubator.
Okay. And do the dip tubes cause any problems with the culture?
So the tubes which go into the flask, they need to be resting in the culture because ultimately liquid needs to be drawn out of the flask, so it needs to pull them out. And the tubes are platinum cured silicone, which is typically used throughout every production process, and there's very little concern about that material negatively affecting cell growth. Because these tubes do sit in the culture during incubation and thus shaking, we also wanted to see if there's any kind of physical damage that might occur from the tubes resting in there. So we did do studies that we looked at cell growth rates again in MyCAP CCX and traditional flasks, but this time we looked at different shaking speeds in order to try to create more and less turbulence in the system around those dip tubes. At the end of those studies, we found that there was no difference, again, in the growth rates, regardless if the dip tubes were present in the MyCAP CCX or there were no dip tubes as is found in the traditional flask.
Okay, so it looks like we have time for just one more question. If we didn't get to your question, go ahead and continue to type those questions in the chat window, and then we will be passing them on to Charles so he can follow up with you directly. So how should the media and inoculum be measured into the flask?
So we recommend, and there was an illustration we had shown on a prior slide, we recommend placing that receiving flask on a balance and then doing those transfers by weight. And in fact, in Sartorius' portfolio of products, we do sell balances as well. And there are applications that are written for these balances to control certain repeated processes. We've made an app specific for this expansion process. And this app and the balance will calculate the volume of media and the culture that's required to be added to the next flask, and then the balance is then connected directly to the pump and will control the starting and stopping of the pump once it's achieved the targeted volume. So effectively, this app automates the cell passaging process to help remove any kind of potential risk of operator error, and also creating good documentation of transfer volumes that are done throughout the passages.
Okay. Well, thank you, Charles.
Yeah, thank you for the opportunity and the chance to present this information. It's a pleasure to be able to tell this story.
And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.